today I'm giving a tutorial on population genetics in the human microbiome. And I know the program says, how do we detect evolution from human gut microbiome data? And that is going to be a big component of what I talk about today. I'm an assistant professor in the Department of Ecology and Evolutionary Biology here at UCLA. So for those of you who are new to the microbiome is comprised of a complex community. It is consisting of hundreds of species of bacteria, archaea, fungi, and viruses all interacting in this community inside of you. And a lot of effort to date has been focused on categorizing who is there and what they are doing. But I've become very interested in understanding how are these species evolving. Now, when we think about evolution of microbes, we think a very famous example of lessons we've learned about evolution of microbes comes from the lab. For example, Lenski and colleagues evolved E. coli in test tubes over thousands of generations and saw repeatable and rapid evolution. So here's that in cartoon form. The bullet indicates an adaptive mutation rising to high frequency. Now, this is a situation where we have evolution in a very simple community, and even though the scientists who created this experiment tried to make the condition as boring as possible, they still saw really repeatable rapid evolution, likely because they were still putting the bacteria in some environment different from what they were used to. So now what happens when we go from this really simple community to one where we have a very complex community like the one in our microbiomes? Here you have all these species, and not only the species, but also the strains within each species interacting. And it could be that instead of evolution, you see species fluctuations over time in response to some selective pressure. And this kind of phenomenon, the shift in species abundances over time, has been documented extensively in the microbiome, and we know this happens in diseased people and after antibiotics, etc. Another way by which the microbiome could respond to selective pressures is by invasion of a better fit strain from the outside environment. So here you have this strain that maybe harbors an antibiotic resistance gene, and it colonizes the gut. But the other way that the microbiome could respond to change is by evolutionary change, where you have a new mutation that enters the population and then spreads to high frequency. And when I talk about this concept of evolution, especially in the context of the microbiome, I want to make clear that I'm talking about a change in allele frequency, not a change in species composition. Now, there's abundant opportunity for evolution in the microbiome with a very high ratio, 1-to-1 ratio of human to microbial cells, combined with the mutation rate and generation time. It is estimated that there are almost 1 billion mutations entering our microbiomes daily. And that's a hotbed of evolution happening right inside of us every single day. So what happens to these new mutations that enter our microbiomes every day? What determines their fate? Well, we can look at population genetics as a way to understand the fate of these mutations. And in population genetics, for any population, we're thinking about drift and migration, adaptation, recombination. And these are the very forces that operate inside of our gut as well. But why should we care about the fate of these mutations? Well, these population genetic processes can actually give rise to a range of traits in the microbiome. So, for example, mutations in our microbiome are responsible for the ability to digest food. There's a gene present at very high frequency in Japanese individuals that actually confers the ability to digest the sushi wrapper seen here. And in mice experiments, people are seeing repeated and rapid evolution in response to high-fat diets. So the gut microbes are adapting to what we eat. And they're playing a really essential role in aiding the digestion of that. Of course, the mutations in our microbiomes are also responsible for antibiotic resistance. And that's, as you know, a really large and huge public health issue. And then another really interesting anecdote about the mutations and their functions in our microbiomes comes from mutations in the species Egertella lenta. Interestingly, it's not the presence of the species Egertella lenta that inactivates this heart drug digoxin. It's actually the presence of a genetic variant in that species that has such a critical impact on host phenotype. So these are just anecdotes of what effect the mutations in our microbiome have on our health. And yet we're spending a lot of time studying species fluctuations, ignoring a whole swath of diversity within. And to really understand how that diversity arises, we need to understand the population genetic processes that give rise to those mutations. Now, what's really special about studying this in the microbiome is not just its relevance to health, but also the fact that all of these population genetic forces are happening in the context of this really complex ecosystem. And so it's not just evolution that's happening, but it's also ecology. So you have ecology, maybe you're counting the distribution of species. In evolution, you're looking at the distribution of allele frequencies, and they're interacting with one another. And that's what makes studying population genetics in the microbiome so special and different from studying it from any other organism or system. I wanted to highlight a review that I actually wrote on this topic, population genetics in the human microbiome. This is with my former postdoctoral advisor, Katie Pollard. And much of what I'm going to talk today about in this tutorial is covered also in this review. I'd also like to highlight several papers that have come out recently on evolution in the microbiome. And so with many different types of technologies, including shotgun sequencing, single-cell isolates, special barcode read technology, high-C technology, people are seeing a similar phenomenon, which is in healthy hosts, people like us any random person who is not necessarily taking antibiotics, doesn't necessarily have a disease, in healthy people, you're seeing evolution happening on daily, weekly, monthly timescales. And so this is a phenomenon that's being documented in many studies across different technologies and platforms. And we're learning a lot about exactly how does evolution operate in this really complicated system. Much of what I'm going to talk about today is based on my own paper, co-first authored with Benjamin Good. And we wrote this paper on evolutionary dynamics of bacteria in the gut microbiome within and across hosts. And in this paper, we started to analyze shotgun data. And I'm going to make a case for shotgun data, despite the marvels of what we can learn from other types of data, because shotgun data is so ubiquitous. If you go on any database, you can download terabytes and terabytes of data. Really awesome data, like spatially resolved data, temporally resolved data, data from hundreds and thousands of individuals. Is a treasure trove of information hanging out right there in front of our noses for free. And if we can learn to manipulate and understand that data, we can make a lot of insights about evolution in the human microbiome. And moreover, I'll make the case that this is a really great system to study because you can think about different hosts as replicate experiments. So we can kind of start to understand general trends. And you can do that especially with shotgun data, given how much data there is from hundreds and thousands of individuals. So going back to this menu, the population genetic processes in the human microbiome that I'll talk about today are drift and migration, adaptation, and recombination. But first, I'm going to add to that menu data, because I know I mentioned shotgun data, but maybe some of you may not be familiar with what that is. So let's dive in. So what kind of data do we need to study population genetics in the microbiome? So I showed you these 3 different processes that can happen in the microbiome. And a very popular and affordable type of data for studying microbiomes is 16S data. So 16S data, if you're not familiar with it, is a kind of a barcode technology. Or amplicon technology. And so essentially, it looks at which gene, this particular gene, 16S gene, and virtually every species has this, and what it does is it picks up on the specific sequence for each different species, and you can, in a very inexpensive way and rapid way, categorize who is there. But the extreme limitation of this technology is that you cannot see what else is going on in the rest of the genome. And that severely limits your ability to look at evolution. So you need a technology that can tell you about what's going on in the rest of the genome. And that brings me to whole genome shotgun sequence data. So what that does is you take a sample, and for those of you who don't know, a lot of the data for microbiome studies comes from stool. So you take a stool sample, you extract the DNA, and basically what we're doing is we're taking all this DNA and sharing it, cutting it into small fragments, and then sequencing that. And what's interesting about this type of data is you get a snapshot of hundreds of species all at once and their whole genome. But this is not easy data to deal with because these reads are only 100 base pairs long. And so it's this really complicated jigsaw puzzle that you need to deal with to be able to figure out what's going on in this data set. It's quite different from, let's say, working with human genetic data where you are dealing with data from one species at a time. So what we can do to simplify this data is to map it to reference genomes. So here I'm just sorting out these reads to different species. And so here is reads going to species 1, 2, etc. And then you can go ahead and take a look at the mutations that are within each species and start to do some population genetics within each species. So for example, you might see this panel of individuals. We could do shotgun sequencing on all of us and look at a common species that's present in us, like Bacteroides vulgaris. It's very, very common. And we can look at a given site, and you'll see in this person's genome, this site is at 80%, this one has something at 30%, etc. And this kind of information is really useful for looking at things like strain transmissions. So for example, we've been working a lot on mother-infant data, and we can track the transmissions of microbiota from moms to infants using SNP profiles. So specifically, we can look for private SNPs that are present in mom and baby only and absent in the broader population. And that's a telltale that mom gave this particular strain of bacteria to the baby. Interestingly, if you look at the number of species shared with the baby over time, you see an increase. But if you look at the sharing of marker alleles over time, you see a decrease. And this shows the power of SNP sharing as a means to really tease out what the source of the microbiome is for the baby. So what this is saying is that while the baby's microbiome is starting to look like mom over time, its source is not actually mom per se. It could be other family members or the environment. Okay, so we talked a bit about data. So now let's talk about some of the population genetic forces and their patterns in the data. Let's talk about drift and migration. So a popular question that has been posed for microbes and other organisms is everything everywhere? And so we can ask how many strains of each species posed in a different way, how many strains of each species colonize a host? So let's take the species E. coli. And I know I'm using the word strain a little bit loosely. What I mean by this is there's a collection of very genetically distinct lineages, and you may get exactly one, a few, or hundreds. And maybe I'll ask you, who thinks that for a given species, let's say E. coli, that you have exactly one strain? Do you want to raise your hand? And who thinks, I see no hands, who thinks that you might have a few strains of E. coli? Okay, I see more. And then who thinks that you might have hundreds of strains? I see a lot more hands. So nobody thinks that you'll have exactly one strain. A few people think that you'll have a few strains. And most of you think that you have hundreds of strains. Okay, so let's take a look and see what the answer is. So we can try to understand this by actually looking at the genetic variation within hosts. And if you had hundreds of strains, meaning if your microbiome looked like everybody else's microbiome and there was no difference between what was inside you and the rest of the world, then everyone's diversity should be similar. The diversity within us should be similar to diversity across us. And we can actually rule that out. So let's take a look at how. So what I'm going to do is I'm going to plot the distribution of allele frequencies for each host. And that's plotted here for 3 different species. 3 different hosts for the species Bacteroides vulgaris. So these are 3 hosts from the Human Microbiome Project. And they are healthy people from North America. And these are what population genetic folks would call site frequency spectra. And so what I'm plotting on the x-axis is the allele frequency, and on the y-axis the fraction of sites. And you can see just by looking at these plots that the distribution of allele frequencies are really different across hosts. It's almost as if like each person has their own personalized signature. And in fact, the genetic diversity in our microbiomes has been called a so-called microbiome fingerprint because it is so individualized and so unique to us. But what are these bumps and how do we make sense of this? What is generating this? And how do we get back to this original question of how many strains of each species colonize a host? So taking a look at this first host, you can see that we have this bump at 15%. And then we also have this mass at very low frequency. And this host has something at 50%, and so on. And what this is reflecting, actually, is the lineage structure within hosts. So this host is actually being colonized by 2 strains, one at 15 and one at 85%. And these bumps are showing up as polymorphisms within hosts, but what they really are fixations that have arisen between these 2 lineages long before colonizing the host. And this mass of sites arises from the growth of a strain or a lineage within a host. So every time you have a population growing, you'll see new mutations spontaneously arising. So we can, by looking at this plot, rule out the hypothesis that we're colonized by hundreds or everything that's in me is in you, because this is not the case. These are highly unique signatures. And moreover, we have these weird bumps that are consistent with actually a few strains colonizing hosts. We can also rule out the fact that exactly one strain is colonizing us because, again, we have these bumps. If we only had one strain, we'd only see these low frequency mutations. So this distribution is fairly interesting. Why is it only a few? And if you notice, it's like on the order of 2 or 3 or some discrete number. It's not 10 or 15. And it's not really clear what ecological force governs which strain gets into you. There is one exception to all of this, and that is the species Bacteroides fragilis, which interestingly only one gets in due to competition. But for the majority of gut commensal species, it's just a few. So is everything everywhere? Well, it could be that, okay, it's not quite everywhere, but could it be that the strains that are circulating in North America happen to also be in Asia? And it just that our own profiles happen to be unique. Well, we can look at the biogeography of gut microbes. This is a paper by Truong et al. And what I'm showing you is data from Truong for Faecalibacterium prausnitzii, E. rectale, Prevotella cropri. Colored in these dots are data from different individuals from different parts of the world. And what you see is actually there is clustering of the genetic identity of these strains from around the world. And so it does seem that the answer to this question, is everything everywhere, is no. So what we have within us shows a lot of structure, and globally there is also a lot of structure. I should also say that some species don't necessarily show this biogeography. Some of them actually are pretty well mixed around the world. And that leads me to some of the open questions on this topic of population structure, which is what ecological and evolutionary processes govern the, I call it oligocolonization of individuals, that is, colonization of one or a few strains, and why do different species show different geographic distributions? Did I see a question?
Yeah, can you return to that three phylogenies over the
Yes.
Yeah, so all these species are over different strains of the same species? Because
Yes.
I don't understand. Yeah, so, and these strains just, so it's, you disregard the geography and then you color by the geography.
Correct. Yeah, so just to repeat the question and to clarify, each node is a different strain.
Yeah.
We disregard the geography, first make the phylogeny, and then color by geography.
And how can you assess how significant the geography by the coloring?
This is not, I have to admit, not my work, and I should know what statistical test they did, but my guess is it is significant. Yes?
Can I ask one more question since Ziggy already interrupted you?
Sure.
For these variant allele frequency plots,
Yeah.
how can you rule out the possibility that it is hundreds of strands, but they're all at less than 1% frequency?
That's a great question. So how can I rule out the possibility that there are hundreds of strains, but they're all at less than 1% frequency? I cannot. And, it's possible that they are hanging out there, but I guess I haven't shown you the full data. And, maybe this doesn't really quite illuminate it, but one thing that we noticed is there's very rarely strain sharing between individuals. It's like super rare. And when there is sharing, it's interesting because they'll be diverged by maybe 10 SNVs, and there'll be patterns of recombination between them. So it's not as if they're clonal descendants of a recent ancestor. And my thinking is that should there have been actually more strains colonizing us just secretly at low frequency, I think we would see a lot more strain sharing between individuals than we do in the data. So that's why I think we don't see that. But this is a great question. And please do interrupt. This is supposed to be a tutorial, so I'm more than happy to help and answer questions as we go along. Okay, so going to the next part, which is adaptation. How do we detect that, and what does it look like in the human gut microbiome? So how do we, so I'm going to use the word evolution, but adaptation is where a mutation is beneficial and can rise to high frequency quickly because it is beneficial. And how do we detect such events within hosts with shotgun reads? Ideally, what we're going to look for are mutations that go from low to high frequency over time. But you can see this is a really complex problem with the mess that this data is. So I've already shown you how we can simplify it by mapping reads to different species. And then we can look for frequency changes within a species over time. But even then, it's still not that easy. Detecting evolutionary events is not so simple because, as you just saw, multiple strains may be colonizing a host at high frequency, and those strains are genetically distinct. And their fluctuation over time can generate allele frequency changes that can confound our ability to detect evolution. And so our solution is to infer individual lineages' genotypes so that you can actually look for evolution on the background of one of the lineages. Put in another way, you want to know the haplotype of a lineage colonizing you so that you can see the genetic change on the background of that haplotype, and you'd expect that future changes would accumulate on top of that haplotype. So we don't want to confuse this with strain fluctuations where changes don't propagate on top of each other. Now the complication is that you only witness the allele frequencies, right? So you've already seen this plot where we're looking at the allele frequencies and we're looking at the lineage structure. And we can leverage these patterns to try to tease out the haplotypes of the most common lineages. So, for example, this host has a very common haplotype at 85%. And it is possible that we can pull out that haplotype because it's so abundant. And other samples, however, have very complicated lineage structures. Like this might have 3 or 4 strains colonizing it, making it difficult to tease out that haplotype. So we're going to only focus our attention on simple microbiomes, or microbiomes with simple lineage structures, such that we can confidently phase that common haplotype and then track evolution. And so here we can do that, as I just mentioned, for this host where you have one haplotype at very high frequency. And so here you have, at let's say 2 sites, a T at high frequency. And you can assign that T to the most common lineage, and in that manner phase its haplotype. But that isn't the case for, let's say, this sample, because as you can see, the 2 lineages are colonizing a host at roughly 50-50 frequency. And there's an equal probability of assigning the wrong allele to the wrong lineage. And that phasing error can actually swamp out your signal for evolution. So we want to focus on those samples where it's easy and we can confidently phase the haplotype, and we've put error bounds on this. And after we do that, we can then look for mutations that accumulate to high frequency on the background of that haplotype that we've been able to phase. Okay, so we asked this question back when we were writing this paper: do bacteria evolve in the host gut? And we developed this approach to phase haplotypes, and we set out to look at this in the Human Microbiome Project data. And just kind of reminding you why this was an important question to ask. Once again, these are healthy people, and we're looking at things happening on short timescales of 6 months. And it's not evident that these bacteria would actually evolve on such short timescales, because ideally what you have in your microbiomes are stable and optimally adapted to you. And in fact, what we see is evidence for evolution. So what I'm plotting is some data for the species, again, Bacteroides vulgaris. And what we noticed was a kind of a bimodal distribution of SNP changes between these 6 months. So here, these 3 individuals show on the order of 10,000 SNP changes. And these individuals show on the order of 5 or 10 SNP changes. And then you have a bunch of people showing 0 SNP changes. So this distribution was quite interesting. It suggests either nothing's happening, or our methodology is not sensitive enough to detect what's happening, or there is some massive turnover of SNPs, or there's just a handful of SNPs changing. And what this is actually pointing to are two processes generating SNP changes in the gut. This is pointing to strain turnover, and this is pointing to evolution. So let me elaborate. So here is a phylogenetic tree describing all the, let's say, individuals in this room and the relationship of Bacteroides vulgaris in our guts. And you'll see that there's actually a very long divergence time separating our strains of Bacteroides vulgaris. And that long divergence time is so long that you'll see a lot of mutations accumulate, separating our strains. In fact, our strains are diverged on the order of about 1%. Or in other words, 1 in 100 base pairs of Bacteroides vulgaris from my gut microbiome, from your gut microbiome, is different. And do you know which other two species are diverged by 1 in 100? Humans and chimps. Can you believe it? We call two strains Bacteroides vulgaris, even though they're so diverged. And when you look at humans and chimps, they're so different and equally diverged. So there's, an interesting thing going on here about what we consider a species. But that aside, you can see there's so many SNP differences accumulating here that's going to generate a lot of changes when a new strain comes in. Whereas looking at very short periods of time, of 6 months, you'd only expect a few changes to happen. And in fact, if we plot the average number of SNP changes within hosts versus between hosts, you'll see about 10,000 SNP changes. That's if you think about the bacterial genome being on the order of 1 million, that's 10,000 divided by 1 million is 1% is different between you and me. And then within hosts, it has this kind of bimodal distribution, which I'm hiding here as just a single bar, but you'll see on the order of either 10,000 or just a few hundred, or just a few. And so we can actually put a line here indicating the mean number of SNP differences between hosts, allowing us to separate what is going on evolutionarily from what is a strain turnover. So we were very excited to see that indeed our bacteria in the gut microbiome can evolve. And since then, we've gone on and applied this methodology to more than 700 infant microbiomes. And we asked the question of how do our microbiomes change over the course of our first year of life? And this is work actually done by Daisy Chen, who was a Bruins and Genomics summer student here at UCLA. And we looked at different time periods over the course of the first year of life of a baby. And we looked at the rate of evolution in the gut microbiome as the baby progressed through that first year of life. And we saw actually a decay in the rate, surprisingly. And so when the baby, when you compare mom and baby at the time of birth, and you treat mom as a time point and the baby as another time point, you see a really rapid rate of evolution. Then, as the baby enters its first week of life, that rate drops, and it continues to drop over the first month and the first year of life. And by the time you reach adulthood, this is what we were measuring earlier in Human Microbiome Project data, you see that this rate drops, and it's almost 100-fold less than what you see at the initial time of birth. Go ahead, please.
So yeah, I looked and I saw that this is logarithmic, the scale, and actually it's really nice because it complies with methylation rates.
Oh, wow.
Yeah, that's amazing.
Thanks for pointing that out. I didn't realize. So
Yeah,
the point was,
it is exponential. Yeah, that's amazing. Yeah.
if we turn this into a log scale or numbers since birth, and you put in log, it becomes a scaling relationship.
That's amazing. Yeah.
I'm not showing data for rate of strain turnover, but we see the same exact trend. And then I'm about to show you what we'll look at, what we see when we see gene change over the first year of life. But yeah, this was really surprising how rapid evolution can be, and points to a lot of volatility in our microbiomes right at birth, and potentially a role for evolution in helping to stabilize our microbiomes, especially in the face of new diets in the baby, a brand new immune system, and also new community members. Everything's really new as a microbe enters. It's under a lot of pressure to figure itself out, and there's just a lot of loss of species, but also turnover of strains, and also apparently evolution. So yeah, oh sorry, I heard some. So we indeed see evolution in the host gut, but that isn't the end of that story. How does rapid evolution impact the ecology of the microbiome? How does adaptation of microbiota impact host health? How much adaptation is there? What are the targets of selection? These are just a smattering of questions that we can ask. And last night actually there was a really amazing paper that dropped in Nature, which showed that there's evolution happening along the spatial gradient of our guts, allowing bacteria to sometimes translocate to different parts of the gut. And it's that evolution that's really responsible for the ability of the bacteria to survive in different environments and even become pathogenic. And so the evolution of our microbiomes is something that continues to be studied actively, and there's just a lot to be discovered. So the last thing I'll touch on is recombination. And this is a really, really important force in population genetics. It's the way by which genetic material is exchanged and keeps the population cohesive. And it actually does play a really important role also in adaptation happening inside of us on relevant timescales of just a few months. So here's a kind of a schematic of how recombination might happen in the microbiome. A lot of you might think, well, these are bacteria, so they're reproducing clonally. When do they have the opportunity to recombine? Well, you can have recombination for sure, and there are different ways by which that happens. First, you need DNA to come in contact with another strand of DNA, so that recombination can happen, and it can happen via transduction, conjugation, competence. And then once the two strands of DNA are together, you need there to be exchange. There can be homologous recombination, where you see two similar strands lining up, and you see exchange. You can have non-homologous recombination, where only parts of the DNA touch and exchange. Homology-facilitated illegitimate recombination, where it sort of touches and it still exchanges. You can also have plasmid integration. And then the last thing is, well, okay, this is all happening, but how do we detect all of this? Well, we could use tools like the 4-gamete test, which basically, if you see at 2 given sites, all 4 gametes present, it violates a simple tree or clonal process generating this pattern, and instead you have to invoke recombination potentially. If you see less than 4 gametes, then there's no evidence for recombination. Put another way, you can measure linkage disequilibrium, which is basically looking at correlations between sites, and you can measure it as a function of genomic distance. And if you see a sort of a decay like this, then you know that you have evidence for recombination. And so what we did in our paper was we took haplotypes from different hosts in our data set, Human Microbiome Project data set, and we measured linkage disequilibrium across those haplotypes, and that's measured in blue. And what we saw was a decay indicative of recombination. And we saw this for most of the commensal gut bacteria species, and in fact, it's a pretty steep decay indicating recombination is a common thing happening in the microbiome. Admittedly, this is data collected cross-sectionally across hosts, but we can also look further and see what's going on inside of us. Do we see evidence for horizontal gene transfer? And going back to the mother-infant data, indeed we do. So what we did was we took shotgun data, we looked for evidence of genes coming in and out based on copy number, and we were able to infer if genes were gained or lost as a function of time in the baby. So this is plotting the gene changes per day, and what you see are some interesting patterns. From the mother-infant transition, you see an elevation of loss of genes. So the stochasticity potentially as the microbiome is moving into the baby is resulting in a loss of a lot of diversity, not just at the species level, but apparently also at the gene level. And for whatever reason, the lineages that are present in mom no longer have all their genes when they reach the baby. And later on, over the first year of life, you see the trend flip, where you see more gains than loss. And we know that the microbiome matures functionally over time. It could be that recombination is a way by which the microbiome is acquiring back the diversity that it lost. And more generally, it seems that recombination plays a really important role in seeding adaptive events in our microbiome. And this makes sense, especially in this ecologically rich context, because why spend the energy and time innovating something you need when it already exists? If your neighbor already has it, just take it up. And so that may be a way by which evolution is distinct in this really ecologically rich community compared to one in a test tube. Of course, there are tons of open questions. Do bacteria recombine randomly with each other? There's this population structure that we've talked about. How do they get in contact with each other in the first place? When do bacteria have the opportunity to recombine if hosts are oligocolonized, right? And how does recombination impact the mode of adaptation? And these are just a few questions, but we barely have scratched the surface of understanding these forces in the microbiome. So I'd like to conclude by sharing with you a few points about population genetics in the microbiome. A genetic variation in the microbiome impacts our health. There's a lot of data. Inter and intra-host population structure exists. Bacteria can evolve in the human microbiome on short timescales. There's extensive recombination, and the rapid evolution in a complex community affords us the opportunity to study eco-evo relationships. With that, I'd like to thank the co-authors whose work went into this presentation: Daisy Chen, Benjamin Good, Katie Pollard, and Oscar Halachek. I'd also like to thank my wonderful lab. They have contributed in many, many ways to stories that I have not had the chance to share with you today. And I'd also like to thank my funding. And with that, I'd be happy to take any questions.